Everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with metabolism of LDL. In today's session, we will continue with the same, in which HDL has three forms: HDL 1, 2, and 3. Then we have functions and clinical significance of HDL. Then we have HDL subfractions. Then we will study free fatty acids. And lastly, we will have cholesterol and beta cells. Good morning everyone. Let us begin the topic. Metabolism of HDL. The intestinal cells synthesize components of HDL and release into the blood. The nascent HDL in plasma is discoid in shape. The free cholesterol derived from the peripheral tissue cells are taken up by the HDL. The ApoA1 of HDL activates lecithin cholesterol acyl transferase present in the plasma. In this diagram, we can see that phospholipids ApoA1, ApoC, and ApoE are transferred from liver to intestines. In the intestine, the LDL synthesized is discoid and nascent HDL, which after the action of lecithin cholesterol acyl transferase converts it into mature HDL. This mature HDL is ready to go to the liver once more. The LCAT then binds to the HDL disc. The cholesterol from the cell is transferred to the HDL by a cholesterol reflux regulator protein, which is an ABC protein. Lecithin is a component of 5-phospholipid bilayer of the HDL disc. The second carbon of lecithin contains one molecule of polyunsaturated fatty acids. Lecithin cholesterol acyl transferase. Lecithin is converted to lysosyl lecithin and cholesterol is converted to cholesterol ester in the presence of enzyme LCAT. It is transferred to the third hydroxyl groups of cholesterol to form cholesterol ester. The esterified cholesterol, which is highly hydrophobic, now moves into the interior of the HDL disc. The reaction continues till HDL becomes spherical and increased cholesterol esters are formed. This HDL particle, designated as HDL3, the mature HDL spheres are taken up by liver by ApoA1 mediated receptor mechanism. HDL is taken up by hepatic scavenger receptor B1. Hepatic lipase hydrolyzes HDL phospholipids and triacylglycerol and cholesterol esters are released into the liver cells. In this diagram, we can see hepatic lipase or HL, which brings about metabolism of HDL, ApoB48 and ApoB100 all together are directed towards the towards the liver in the presence of enzyme hepatic lipase. The cholesterol that reaches the liver is used for the synthesis of bile acids are excreted such in bile. The scavenger receptor B1 is identified as HDL receptor with a dual role in the HDL metabolism. Its first role is in liver and steroidogenic tissues. It delivers cholesterol ester to the tissues, whereas its second role is in the tissues and other tissues. It is involved in reverse cholesterol transfer. When the HDL3 remains in circulation, the cholesterol ester from HDL is transferred to VLDL, IDL, and LDL by the cholesterol ester transfer protein. This will help you to relieve product inhibition of LCAT so that more cholesterol can be taken up. Triacylglycerol from VLDL, IDL, and LDL is transferred to HDL is in exchange for the cholesterol ester. In this diagram, we can see that HDL is converted to VLDL, IDL, and LDL with the addition of cholesterol in the presence of protein. HDL ester cholesterol ester transfer protein, and VLDL, IDL, and LDL is converted to HDL with the addition of LDL triacyl glycerol in the exchange for cholesterol ester. The HDL particles that are rich in triacylglycerol and spherical are called HDL2. These particles are first acted upon by hepatic triglyceride lipase before being taken up by scavenger B1 receptors in the liver. The efflux of cholesterol is from peripheral cells to HDL is mediated by ABC transporter protein. In the diagram we can see peripheral cells converted to HDL are and with the removal of cholesterol is by ABC transport protein. The reverse cholesterol transport to the liver through HDL needs the activity of LACT, CTP, and ApoD. The functions of HDL. HDL is the main transport form of cholesterol from peripheral tissues to liver, which is later excreted through bile. This is called reverse cholesterol transport by HDL. The only excretory route of cholesterol from the body is the bile. The excretory cholesterol needed its prior esterification with polyunsaturated fatty acids. Thus, the PUFA will help in lowering the cholesterol in the body, so PUFA is anti-atherogenic. Coming to clinical significance of HDL. The level of HDL in serum is inversely proportional to the incidence of myocardial infarction. As it is anti-atherogenic or protective in nature, HDL is known as good cholesterol in common parlance. HDL level is less than 35 mg per deciliter increase the risk, while its level more than 60 mg per deciliter saves the person from coronary artery diseases. Here we can see good cholesterol. Bad cholesterol stores cholesterol in the bloodstream, whereas good cholesterol regulates HDL storage and promotes excretion. Coming to HDL specific receptors. The main receptor for taking up HDL is the scavenger receptor B1. The liver is receptor binds the HDL with the help of ApoA1. Then cholesterol is internalized, but not the HDL particle and ApoA1 are not taken up. In the tissues, the SRB1 accepts the cholesterol into the HDL. Coming to HDL-related transport proteins. The reverse transport of cholesterol from tissues to liver needs two transporters, ABCA1, that is ATP binding cassette transporter A1, and ABCG1. The ABCG1 is helping the transporter of cholesterol from cells to HDL, whereas ABCA1 helps efflux of cholesterol into ApoA1 in the destroyed HDL. Coming to HDL subfractions. HDL1 or HDLC is seen in animal fed with cholesterol. It contains ApoE1E. HDL2 is definitely good and its level is inversely proportional to the atherosclerosis. It is again subfractionated into HDL2A and 2B, and HDL2B is main anti-atherogenic factor. ApoA1 is maximally present in HDL2B. HDL3 is fractionated further into 3A, 3B, and 3C. It contains ApoA2. It also contains ApoD, which is lipid transfer protein. By the action of LCAT, more cholesterol is trapped into the nascent discoid HDL, as we have seen in the diagram earlier. As more and more cholesterol is accepted, the particle size is increased and HDL is generated. As further cholesterol is taken up, particle size is further increased and less dense HDL2 is formed. The interchange of H2, HDL2 and HDL3 is called HDL cycle. Apolipoprotein J or ApoJ is the glycoprotein associated with HDL2. It is synthesized by foam cells in atheromatous plaques. Lipoprotein X is a lipoprotein with cathodal migration seen in patients with obstructive jaundice. In this diagram, we can see chylomicrons and VLDL give pre-beta H1 HDL1, which is transferred to with the help of enzyme glycine cholesterol acyltransferase to HDL3 to HDL2 and finally to HDL1. HDL2 contains ApoA2, whereas HDL1 contains ApoA1 and ApoE. Free fatty acid. It is also known as non-acidified fatty acids. It is complexed with albumin in plasma. The free fatty acid derived from lipolysis of triglyceride stored in adipose tissue by hormone-sensitive lipase. In the diagram, we can see triglyceride is converted into FFA in the presence of enzyme hormone-sensitive lipase. Free fatty acids may be long-chain saturated or unsaturated fatty acids. The FFA molecule transported to heart, kidney, and skeletal muscles and liver and other tissues. The free fatty acids are either oxidized to supply energy or are incorporated into tissue lipids by esterification. In the tissue cells, free fatty acid albumin complex is dissociated and FFA binds with lipid acid transporter protein. It is a co-transporter with sodium. After entry into the cell, the FFA is bound to fatty acid binding protein. The half-life of free fatty acids in plasma is very short, that is 1 to 2 minutes. During starvation, about 40 to 50% energy requirement of the body is met by oxidation of free fatty acids. In this diagram, we can see that fatty acids combine with fatty acid binding protein and its function is with ribosome, mitochondria, nucleus, endoplasmic reticulum and lipid droplet. The blood level of free fatty acid is very low in the fully fed conditions, high in the starved state and very high in uncontrolled diabetes mellitus. Coming to cholesterol and beta cells. The accumulation of cholesterol in the beta cells causes perturbations in glucose metabolism, reduces insulin secretion, and can be associated with a diabetic phenotype. Cholesterol is also key determinant of beta cell membrane organization and cell survival. The ATP binding acid transporter A1, which effluxes cholesterol from lipid-free or lipid-poor apolipoprotein A1, which is the principal apolipoprotein in HDLs, and it is crucial for maintaining beta cell or cholesterol homeostasis and functions. It is also suggests evidence suggesting that different lipoprotein classes have varying effect of beta cell functions and survival. It can cause beta cell loss if allowed to accumulate in the cell in an unregulated manner. The maintenance of beta cell cholesterol homeostasis therefore is important for preventing the onset of insulin and the development of type 2 diabetes mellitus. Here ends the session. Kindly like, share and subscribe the channel. Thank you.